Hello dear students. So in our previous lecture we have finished microbiology of food. So in today's lecture we are going to start factors affecting growth and survival of microorganisms in food. But before starting this topic, we'll see some basic regarding growth and reproduction in case of bacteria. We'll just recall that basic. We're not going to take much details regarding growth and other growth related parameters in case of microorganisms. So growth is an orderly increase in quantity of cellular constituent. This is a basic definition of growth, that it is an orderly increase in quantity of cellular constituent, and it depends upon ability of a cell to form new protoplasma from nutrients available in environment. So in most bacteria, growth involves increase in cell mass as well as and number of ribosomes, then duplication of bacterial chromosome, synthesis of new cell wall and plasma membrane and other cellular components. And these are the basic events that normally occur during the growth of bacteria. So growth in case of bacteria means simply it is increase in numbers. So what is meant by growth in case of bacteria? Increase in cell numbers. Otherwise, if you see other than bacteria, the living things other than bacteria, in case of that the growth is increase in cell mass as well as cell size. But here in case of bacteria we define growth as increase in number of cells. So bacteria, they mostly divides by binary fission. It is a method of asexual reproduction and it is mostly found in bacteria. Sorry for the interruption. Okay, so what is meant by binary fission? So it is nothing but when once two cells they are divided from one cell, so that process is known as binary fission, and this is a sexual mode of reproduction. So for unicellular organisms such as bacteria, growth can be measured in terms of two different parameters, that is changes in cell mass and changes in cell numbers. So as I have told you earlier, that the growth in case of bacteria means increasing their cell numbers as well as cell mass. So there are several methods by which we can measure cell biomass. So these are direct physical measurement by dry weight, or you can take direct volume of cells by centrifugation. Then you can estimate the growth by chemical measurement. There are some of the chemical compounds like total nitrogen, total protein or total DNA. Then you can measure chemical activity. Then simply by measuring turbidity you can again calculate or observe growth. So all these are, we're not going to see this topic in much more detail, okay, because this topic you have already studied. So there are some methods for measurement of cell numbers. In case you can count the cell numbers by direct microscopic count, and then there are some electronic counter chambers also present. Then you can count the cell numbers by indirect viable cell count. Then bacterial growth curve. So there are four phases present in bacterial growth curve. These are lag phase, log phase, exponential phase, or log phase, or stationary phase, and death phase. So you are all familiar with these phases. We will not going to study in detail. So these are four phases through which bacterial growth occurs, and this is particularly this growth curve is designed from their growth pattern, and these are four phases. And generation time, for the generation time is the doubling time which requires for one cell to divide into two cells. So this is again different for different types of bacteria. For E. coli it is approximately 15 to 20 minutes, and you are all familiar with the concept of generation time and growth rate. And these are examples of several bacteria and the medium on which they grow, and their generation time is given in the right side of this table. And this is formula through which we can calculate generation time. So all these are basic things. We will not going to study much more details. We have to focus on the next unit or next point, that is factors affecting the growth and survival of microorganisms in food. So we all know that microbial growth is pretty much affected by several factors like pH, temperature, then water activity. So how these factors affect the growth of microbes in particularly food. So in this point we have to cover details regarding this all these factors. So basically food spoilage means the original nutritional value, texture, flavor, etc. of food are damaged. So what actually food spoilage is that? So it alters the nutritional quality of food as well as its structure, then its flavor, and foul smell or off flavor get developed after spoilage of food. So these are basic criteria to define spoiled food. Means what? The nutritional properties get changed, then texture get changed, then flavor get changed, and it produces off flavors. Okay, of damage the food. So these are all criteria for spoilage, and we call this food as damaged food, and food become harmful to people and unsuitable for consumption or eating. So food can deteriorate as a result of two main factors. First factor for spoilage of food is growth of microorganisms. Obviously the microbes produces some components like toxin, and this toxin produces harmful effects after ingestion of such a contaminated food, and usually they contaminate surface of that food. So usually from surface contamination, and especially important in processed food. So these kind of micro, usually in the daily basis what we eat cooked food or freshly prepared food, so that the chances of microbes in that food are very minimized, but you can find the growth of microbes in some processed foods or food which is kept for a longer time. But the food we eat for 2 or 3 times in a day, that is freshly prepared and it is well cooked, so there are rare chances of occurring of microorganisms in that type of food. So mostly microorganisms they are present in processed foods, just like the food that is packed in some containers and other means. Then next, so first reason for spoilage of food is that growth of microorganism, and the second is the action of enzymes. So from within the cell, a part of normal life processes, so responsible for respiration. For instance, it is important to note that many plants that are fresh vegetables and fruits are still alive when brought, and even when eaten raw, and meat from animals undergo gradual chemical changes after slaughter. So as the things we eat they are derived from some living matter, just like meat or vegetables. So their intrinsic enzyme activity alter their internal characteristics. For that, will get means it get undergoes gradual chemical changes after processing or normal its cutting, or you may call it chopping and other processes. So these two things, that is growth of microbes and action of enzymes. So these are two factors that are mainly responsible for spoilage of foods. And in this particular table you can see a primary source of microorganisms that found in foods. So these are the primary sources through which different kinds of microbes, and in that especially pathogenic microorganisms, they enter into that food stream and they causes spoilage of food as well as other food related products. So the various sources through which microorganisms gain entry into the foods, they are listed in this table. So first is microflora present in soil and water. So you know soil and water, they are the largest reservoir of microbes, and through the soil and water microbes can easily transfer to food stream. So these are the main sources from where microbes can enter. Then microflora present in air. Here also we carry out all the process of food, just like cooking and its cutting and other things, and its washing in open air. So definitely Microbes can enter through air into this food stream. Then microflora present on plant and plants products. So daily basis we use vegetables of different kinds, and that on the surface of that vegetables, though as they are plants, different kinds of microbes are present. And if the vegetables are not properly washed and cooked, or if that vegetable eaten on a raw basis without any kind of processing, then definitely that microbe will enter into that food stream. Then microbes present in food utensils and equipments. These are again important source of entry of pathogens and bacteria or other microbes into main food stream. So the vessels that we are using for cooking, as well as storage, as well as processing of different kinds of foods, they may contaminate the food stream. Then next, microflora present in animal feeds and microflora present in animal hides. So again, these are different sources. So basically these are sources that are mainly responsible for spoilage of milk, as well as milk related other products, and some meat products also that are derived from animal feeds. And last one is the microflora present in intestinal tract of human and animal food handlers. So overall these are different sources through which microbes, and especially pathogenic microorganisms, they enter into the food stream and they cause spoilage of food. Okay. So this is some table that is showing examples of different types of food, and the type of spoilage that occurs in that food, and the causative organism or the microbes which is responsible for that spoilage of food. So this is table. In that first you can see the bread. So bread is mostly spoiled by different types of molds. That's why it is moldy. And type of spoilage that occur in bread is moldy and ropey. So these are textures you can simply observe by your eyes. And main causative organisms responsible for spoilage of bread, these are Rhizopus nigericus, then Penicillium species, and Aspergillus niger. So basically bread is mostly spoiled by fungi, but not by bacteria, very few times, but almost in all cases bread is spoiled by fungi. And you can see moldy and ropey structure on this texture on the bread, and it is a symbol of spoilage. Okay. Then maple sap and syrup. So you can see ropey, yeasty, pink, moldy. So all these are features from which you can judge this maple sap and syrup, that whether it is spoiled or not. So these are signs of spoilage or types of spoilage. And this maple syrup spoilage is mainly caused by enterobacter aerogens, then different types of Saccharomyces species, then Zygosaccharomyces. And these colors, actually these are colors that are caused, types of spoilage, these are caused by different types of organisms. Say for example, a ropey color or ropey texture, it is due to enterobacter aerogens. Then yeasty texture or yeasty type of spoilage, it is mostly caused by yeast, that is Saccharomyces and Zygosaccharomyces. Then pink texture, it is called by Micrococcus roseus. And moldy means molds, it is obviously it is caused by molds, that is Aspergillus and Penicillium. Then next, there are fresh foods and vegetables, they are spoiled, and you can observe the spoil by simply observing their texture. You can find soft rot, as well as grey mold rot and black mold rot. So these are appearances from which you can judge whether the fruit and vegetable they are spoiled or not. So soft rot spoilage are mainly caused by Rhizopus and Erwinia species. Then grey mold rot, they are mostly caused by Botrytis species. Then black mold rot, they are mostly caused by Aspergillus niger species. So these are the main causative agents that mostly responsible for spoilage of fresh fruits as well as vegetables. Then pickles and sauerkraut, they are mostly spoiled by Rhodotorula species, and you can find film of yeast and pink yeast on the surface. Then fresh meat, which is mostly spoiled by putrefaction process. Putrefaction process is the conversion of proteins to some other components, and it is mostly caused by alkaligens, then clostridium, then protein vulgaris, then Pseudomonas fluorescens, and the meat is mostly spoiled by different kinds of bacteria. Then cured meat, it is mostly spoiled by aspergillus, then rhizopus, then penicillium, and some of the lactobacillus as well as leuconostoc species. Then fish and fish related products, they are spoiled by again Pseudomonas and alkaligens. Then eggs, they are mostly spoiled by Pseudomonas fluorescens as well as other species of Pseudomonas and alkaligens, as well as some species of protease. And these are features through which we can you can observe spoilage that are green rot, then colorless rods, and then black rods, then concentrated orange juice. You can simply observe its spoilage by off flavor, so it produces foul smell, and this is mostly spoiled by lactobacillus, then leuconostoc, then acetobacter, and poultry products, they are mostly spoiled by Pseudomonas and alkaligens. So this is the very informative table, and it is we got very information that different types of foods and their types of spoilage, and which microorganisms they are responsible for this spoilage. Okay, so this table is related to microorganisms that are involved in food spoilage. So these are all foods that are different than other canned foods. So and this is overall profile of food spoilage and their microorganisms. So a variety of intrinsic as well as extrinsic factors determine whether the microbial growth will preserve or spoil the food, as shown in table 3.3. So these are the two factors that are intrinsic and extrinsic, means internal and external factor decide, and these factors actually responsible for growth and spoilage of different kinds of food. Growth in case of microbes, so these factors decide the growth of microorganisms and spoilage of foods. So these factors, intrinsic or food related parameters are those parameters of plant and animal tissue which are inherent part of this tissue. This intrinsic factor includes pH of the medium, then water activity, then redox potential, then nutrient content, and antimicrobial constituent and biological structures. These are the intrinsic factors or internal factors that are responsible for growth of microorganism as well as spoilage of food. And then there are some extrinsic or environmental parameters that are or properties of storage environment which affects both food as well as microorganism, and these includes temperature of storage, then relative humidity, and storage of environment and concentration of gases in that environment. So this is again a table. This is very informative, and this is the actual point of this topic, that factors affecting growth and growth of microorganism and spoilage of that food. So there are some intrinsic factors you can see: nutrient content, then pH, then redox potential, then water activity, then antimicrobial substance, constituent, and barriers. Then there are some external environmental factors like temperature, relative humidity, gaseous atmosphere, growth rate, and implied factors such as synergism, antagonism, commensalism, and proceeding factors just like irrigation, washing, slicing, pasteurization, packing, etc. So all these factors we have to study in much more details. So in next lecture we'll study these factors in pretty much detailed manner. So for till now, all these details in detail we'll see in next lecture. Thanks for watching this lecture. Thank you.